This is Johan, this is Patricia, and I'm Kyle. We're going to give you a presentation today on
Neuroplasticity!
I don't know. Yeah!
something like that. We'll see how this goes. Hello, friends. Our names are Kyle Majewski.
Patricia Petrowska.
And Johan Krumov.
We will be analyzing practice-induced plasticity of the motor system in addition to how physical training influences the mental rehearsal on a practice task, such as leisure golfing.
The study we will be examining today is entitled The Effect of Leisure Activity Golf Practice on Motor Imagery, an fMRI study in middle adulthood. This study was conducted by Ladina Benzola, Susan Meralt, and Lutz Janke from the University of Zurich in Sweden and was published in March of 2012.
Other recent studies have shown that motor skill learning indicate that training-induced structural and functional adaptations occur across the whole lifespan. The article's purpose was to explore these processes in a group of middle-aged adults ranging from 40 to 60 years old.
Previous studies on motor learning have recorded neural responses to various kinds of finger movements. However, this study investigates more complex movements that include several joints, muscles, and different body parts.
This was done by the participants imagining themselves performing a golf swing. This study is important because it can demonstrate that training-induced functional plasticity is not a process limited to young adults, but provides evidence that brain adaptations due to skill learning can occur across the whole lifespan.
The study used 22 volunteers that were on average 51 years old. There were two experimental groups. The control group, who had no golf experience, which consisted of 9 females and 2 males, and the independent group of novice golf players that also consisted of 9 females and 2 males.
Each member of the control group had a counterpart of the same age, gender, and handedness in the novice group. Only the novice group participated in the 40 hours of leisure golf, which was this study's independent variable.
This longitudinal, quasi-experimental study had 2 measurement time points, before and after the novice group completed their 40 hours of leisure golf, and would be considered a between-groups design.
A kinetic motor imagery task was given to all participants at the 2 time points. The participants were shown videos of a professional golfer performing golf swings that would prompt them to imagine their own swing for the same duration of the video clip.
Each participant watched 4 clips of the video sequence. The participants were then asked to imagine performing a golf swing from a first-person perspective while in an fMRI. The participants marked the beginning and end of their visualizations by pressing a button.
The whole brain anatomical images obtained from the fMRI were comprised of 160 sagittal brain image slices. The functional data acquired was comprised of only 34 slices. All participants were asked to rate the tactile and rhythmic difficulty of the imagery task using a 7-point rating scale, as well as the spatial difficulty using a 6-point scale.
The researchers hypothesized that the golf novice group would show a reduced hemodynamic response in the brain regions associated with performing the motor imagery task after having completed the 40 hours of golf practice. This would reflect that less neurons are being activated to perform the same mental task, resulting in a more efficient neural network.
The researchers found no significant difference between the participants' first and second self-reported difficulty scores regarding the mental imagery task. However, even though the participants did not report a change in difficulty of performing the task, participants in the novice group showed less neural activity in the right and left dorsal premotor cortices after they had completed the 40 hours of leisure golf. These significant changes were not found in the control group that did not participate in the 40 hours of golf.
The findings of this study helped to confirm the locations of certain brain regions that are activated during the mental rehearsal of a task that had already been identified in recent studies. Also, one can make the conclusion that physically rehearsing a task, such as a golf swing, can facilitate an increase in neural efficiency due to the fact that less neurons need to be activated to complete said task. Repeatedly rehearsing a behavior can cause the human brain to learn or possibly create more efficient neural routes that are responsible for human behavior, which reinforces the old and common saying, the more you do something, the better you get at it.
One particular strength of the study was that each member of the control group had a counterpart of the same age, gender, and handedness in the novice group. This was an attempt to make a between groups quasi-experiment more like a within group study.
As for the weaknesses, although the authors claimed the middle-aged adults they examined showed the same amount of change in neural activity as the younger participants studied in other experiments, this study could have benefited from examining participants in other age groups to see when neuroplasticity begins to decline. Also, the researchers could have chosen to look at an array of behaviors that varied in complexity, so they could see if the task difficulty affects the participants' ability to undergo a neural change.
Finally, the self-reported measures of task difficulty were not empirically tested before their implementation in this study. Because of this, the responses provided by the participants might not actually reflect the actual amount of difficulty felt, which is a common problem found in most self-reported measures.
This concludes our presentation on neuroplasticity. So if you have any questions, please ask the in-person versions of us. Thank you and have a nice day.